Now that we've talked about the structure of DNA, we're going to talk about DNA replication, which is a fancy word for how DNA makes copies of itself. So the first thing that's going to happen is that the DNA is going to split in half. Remember how the bases in the middle are connected with hydrogen bonds. So you have like A connected to T, remember, and G connected to C. So it turns out, remember, that these hydrogen bonds are very, very weak and they're easy to break. And so when DNA goes to make a copy of itself, what's going to happen is these hydrogen bonds will break and the two strands are going to separate. And this model, the way that this is going to work, is called the Semiconservative Model. And it was actually discovered in a very famous experiment, but we're not going to get into that. So what's going to happen is, if you imagine that this is my original DNA strand, right? In DNA replication, this is going to open up like this. And now you're going to get new bases added to the other side. So the reason it's called semi-conservative is because each new DNA that's made, so now we have two separate DNA strands, 1, 2, and each one consists of one original strand from the original parental DNA, so to speak, and then one new strand that was built base by base, or nucleotide by nucleotide, using special enzymes. So we're going to get into the details of that in a minute. And remember, since the bases only pair up a particular way, if this side was an A, the enzymes will know that the match has to be a T. If this side was a G, and the enzymes bring a nucleotide for the other side, they're going to have to bring a C. So as long as you have one side, enzymes will know what base to put on the other side to build the matching new strand. And so that's what this is sort of giving you an overview of. So this is kind of showing it. So they colored the parental DNA blue. See how when it opens up, A can only connect to T. So enzymes will bring in a T nucleotide and will attach it. And then a G will come and attach to this C, and a C will go to this G, and then a T will come for this A, and an A will come for this T. And in the end, you'll have two identical DNA strands, and each one consists of one original strand and one brand new strand that was built from fresh nucleotides. Now, why does DNA need to replicate? Let's just take a quick step back. Remember a couple chapters ago we talked about the cell cycle. We talked about G1, S, G2, and then mitosis or meiosis or whatever, cell division. Well, S, that is where replication happens, synthesis. So this is where, remember we talked about how the chromosomes look like this, and then after S, they look like X's because the DNA has made a copy of itself. This is how it does that. So this is replication. That's what we're talking about here. This is another picture, again, showing the same thing. We have an original. Notice how the helix untwists, and we get two new strands. Now, it doesn't technically fully break apart at one time. It actually does unzip, and there's some names of some of these parts. This area right here where it's unzipping, this is actually called the replication fork. And so bases are going to be added as the DNA unzips. It doesn't actually separate completely and then the new bases come in. They're actually being added while the DNA is unzipping.
DNA replication, an overview. In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. Here's a simplified close-up of replication, showing what happens along one of the template DNA strands. Using a supply of free nucleotides, DNA polymerase assembles a new DNA strand along the template strand, following the base pairing rules. Adenine pairs with thymine, and cytosine pairs with guanine. Now let's look at the DNA replication process. Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we've untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical daughter DNA molecules. Now, let's review the synthesis of DNA by assembling a new DNA strand. We'll use the top DNA strand as the template and choose from the free nucleotides below. Note that D stands for deoxyribose and R stands for ribose. We're only using the deoxyribose nucleotides because we're building DNA, deoxyribonucleic acid.
Okay, so that was a bit complicated, and they added some extra things, so let me explain the things that they talked about that I didn't talk about yet. First of all, the name of the enzyme that actually adds the bases is called DNA polymerase. And one way to remember this is DNA is a polymer. It's a chain. So polymerase, ACE, remember, I don't know if you remember from the beginning of the year, but if anything ending in ACE is usually an enzyme. Enzymes help with reactions. So the enzymes are actually bringing in and attaching the A, T, G, and C to the old one and building a new DNA polymer. Now it turns out they talked about this thing about antiparallel. If you remember, we really simplified that in class. One side of the DNA has the little pentagon-shaped deoxyriboses facing up, and the other side of the DNA has them facing down. The side with them facing up is sometimes called the 5' to 3' side, and the side with them facing down is called the 3' to 5' side. So let me see if I can, sorry, my pen stopped working here. Here we go. Okay, so this side with these facing downward, this is called the 3' to 5' side. Now it has to do with numbering the carbons in the deoxyribose, and I'm not really going to get into that, but if you come back and take AP Bio, you can learn more about the numbering of the carbons. But here's the bottom line. So it turns out that one side, this is called being anti-parallel, and one side is sort of upside down compared to the other. And that's really the only way that these bases will connect properly. Okay? So one side sort of faces up, one side faces down. So it turns out if we just imagine the DNA unzipping, even though it really does it in these bubbles, when the DNA unzips, it turns out that this enzyme only has the ability, this is my original 5'3", this is my 3'5". The enzyme can only add bases in the 5' to 3' direction. In other words, the enzyme comes in here, this is polymerase that's going to build the new side, and it can go this way. It can add bases in this direction, but it can't add bases this way. This would be building 3 to 5, the match. So bottom line, interesting thing, this side gets copied towards the replication fork. So this is where this is opening up. This is what we call the replication fork, where the DNA opened. The other side doesn't. It gets copied in little pieces. In other words, kind of smart thing on DNA's part, we got two options here, right? We could either wait until the entire thing unzips, because this side can be copied in order. The other side, we have two options. We can either wait till the whole thing unzips and then copy it in the correct way. This way, it would have to be copied. Or, why not, just as it's unzipping, copy it in little pieces. So at one point, in other words, it's unzipped up to this point, let's just say. So I'll just copy this little section. And then now it's unzipped a little further, I'll copy this little section. And then it unzips a little further and I'll copy this little section. And so one side is copied in a straight continuous line. The other side is copied in these little pieces, which happen to be called, they don't mention it in your chapter, but they're called Okazaki fragments. The thing is, once this is completely finished, what you're going to be left with is, now my DNA is completely separated. Well, this one was built in order in one shot, right? So this is all connected. The other one was actually built in little pieces. So there's these little gaps where these primers were attached. And you basically have to fill in these gaps and connect these together. And that's done by an enzyme called DNA ligase. It's like a glue. And what it does is it finishes sealing these gaps so that this side also is one continuous strand. So it's a bit confusing, but in general, just understand that the DNA opens up or unzips. One side runs in anti-parallel to the other, and it turns out that one side can sort of be copied in order. The other side has to be copied in little pieces called Okazaki fragments. You don't have to know what they're called. The enzyme that builds the new chain is called polymerase, and the enzyme that seals these little fragments together is called ligase. Ultimately, the simple version, you end up with two new DNA strands, each one consisting of an original parental strand and a brand new one. This is showing the little replication bubbles they were talking about, and how ultimately, by the DNA getting copied in little pieces here, in the end, it can be faster than if the DNA just had to unzip at one end. You could only copy it, at a certain speed. But if we could copy it in multiple places at once, we can copy a section, a section, a section. This is not actually showing the part about how the strands are anti-parallel. This is showing it a little more. So this isn't drawn overly well because technically here where I made that little circle, there's actually an oxygen at that spot. And I think that would make it a bit clearer if they had actually drawn in the oxygens. That the oxygens are actually face up on this side, and the oxygen on this side is actually here, and the oxygens are face down, because this makes it hard to tell that they're anti-parallel. But they actually are, where one side, the sugars sort of are flipped upside down to the other. And the names 5' and 3', you're not going to be tested on it, but it comes from numbering the carbons, which is sort of something you learn to do in organic chemistry. It's kind of a high-techy kind of thing. But it turns out in a deoxyribose molecule, if you were to draw it, each of these is a carbon. It's another lazy thing they do in organic chemistry is they don't actually put the letter C for carbon. You're just supposed to know that a bend is a carbon. So this is what it actually looks like in deoxyribose, and they didn't even draw the hydrogens and some of the other parts that are here. But you start numbering the carbons in this ring to the right, or clockwise from the oxygens. This is carbon 1, 2, 3, 4, and this is carbon number 5 where the phosphate gets hooked on. And carbon number 3 is where the phosphate gets hooked on the bottom. And the base comes out over here at carbon number 1. On this side, they have it upside down. It looks like this. So this is where the base is attached. And this is carbon number 1, 2, 3, 4. And then the 5th one is actually out here. And this is what attaches to the phosphate here. And this carbon attaches to the phosphate here. So it's kind of complicated. But bottom line, they're anti-parallel. Here's another picture showing, so one side can be copied in order because it's building in the 5'3" direction. The other side has to be copied in these little pieces. The pieces are actually called Okazaki fragments, which they didn't tell you in your book, but you would learn it later. And then this enzyme ligase will come up and seal these gaps. And the reason there's gaps there, they also don't really explain to you, is that in order for primase to know where to start, you have to have a little primer here. And then when the primers leave, you're left with these gaps. This is just a bigger picture of the whole thing. They call the one copied in order the leading strand because it's faster. It's going towards the fork. This is called the lagging strand since it's copying in little pieces. So leading strand
Let's
and lagging strand.